It's my pleasure to introduce Jim Haxby.
Thanks, Luke. There, the mic should be on now. Good to see you. It's great to have the second session of the Mine Summer School, and it's real honor to be the lead-off speaker here. So this is a talk I've been giving many times in different places. This is worthless, by the way, here because that screen absorbs the red light. So I'm going to try to use this pointer. Can you see that little arrow? Okay. And I keep changing the title a little bit and augmenting the talk with new material. And what I want to emphasize today is that in this project that we've been working on for, oh, this is green? Oh, look at that. Okay, I don't have to do this. So that we've been working on for nearly 10 years now. I started with my first graduate student at Dartmouth in the fall of 2008. What we've kind of learned what we're doing as we continue to work on it, and we're really finding now that what we're really making a model of is the information that's encoded in fine-scale cortical topographies, and that we're showing how much this information is shared. As we call this a deep structure, because the information is what we're interested in and what the cortex is doing in cognition. And these fine-scale topographies are the surface structure, and they're idiosyncratic across subjects. There's some principles that are shared across brains, but we don't really know how to align these in terms of cortical topographies. What we can do now is we can align the information that's encoded in these cortical topographies. Now, I hope what I just said is going to make some sense to you as I go through the talk. So first, I always start by acknowledging that the work is really the work of a sequence of graduate students and postdocs. And I especially want to highlight Swaroop Guntapalli, who was that first graduate student in 2008 who really did all the work coding the hyperalignment algorithm. Sam Nastase, here he is having un caffè liscio, an espresso on a street in Rovereto where I found him. And then Fei Long. And is Fei Long going to be? Oh, Fei Long's right there. Okay. Fei Long, who's my current graduate student, and I'll be talking about his work at the end of the talk. And then I hope you all met Jarek yesterday, who was just a central player in computational neuroscience and open neuroscience at Dartmouth. And then we've been working with a group of electrical engineers at Princeton University, where I was before I came to Dartmouth, headed by Peter Ramage, who was the chair of electrical engineering. And then we burned through a sequence of graduate students. I've got Cameron Chen on the end here, but there's actually another person that I have to find a picture for, but I haven't met her yet. Her name is Hejia Zhang, is that right? Hejia Zhang. And we should be meeting her this fall, I hope. So this is an outline of the talk. And first I'm going to just talk about what is the problem. The problem is that the topographies in cortex have coarse-grained structures. So these cortical areas, like there's visual areas V1, V2, V3, auditory areas, category selective areas for faces and places and things like that. But then there are fine-grained topographies that have a spatial frequency that's finer than that of cortical areas. And so can we integrate these into a common model? And so, as I said, we have a different conceptual framework. So instead of trying to align the topographies in a cortical space, we try to align the information in a high-dimensional representational space. And it's actually not just representation. And then I'll talk about how hyperalignment, what we developed so we can derive a common information space with individual transformation matrices, and then validation, a newer version that we just published this year that's based on functional connectivity, and then Fei Long's work on examining individual differences in the fine scale in cortical architecture. So let's start here. So this is, these are some results from a couple of papers that we published about 6, 7 years ago. It was Andy Connolly's experiment. He had subjects looking at 6 different species of animal, 2 species of insect, 2 species of bird, and 2 species of primate. And we measured their brain activity. And in this particular analysis, we looked at a region in ventral temporal cortex and looked at the patterns of response in ventral temporal cortex and asked, could we tell which species they were looking at from the pattern of activity in the ventral visual pathway? And this is a confusion matrix. So the way this works is the input is on the vertical axis, and the classifier output is on the horizontal axis. So when they're looking at ladybugs, how often does the classifier say the pattern in ventral temporal cortex corresponds to the ladybug pattern? Or how often does, and this is Luna moths, and here we have squirrel monkeys. How often does it say it's a squirrel monkey? But then the off-diagonal colors are how often are mistakes made. So if it's a squirrel monkey, how often does the classifier say it's a ring-tailed lemur? Okay? And so the color, you want nice warm colors here, because those are nice high classification rates, and you want cool blue colors here, because that's a low rate of errors. And so you can see this is a very pleasing classification matrix that we were able to classify all 6 species from each other at much better than chance. So the problem is that when we build a classifier like that, we do it separately for each individual subject. So we have to find out, determine what are the patterns associated with Luna moths and ring-tailed lemurs in an individual brain to classify left-out test data. But those patterns in one subject do a terrible job classifying patterns in other subjects. So if we just align the data based on anatomical markers, landmarks, and then try to do a classification where we build the classifier on some subjects and then test it on a left-out subject, the performance crashes. So this is the confusion matrix for between-subject classification, and the diagonal is now mostly blue, and the overall classification goes down from 68% down to about 37%. So that just doesn't work. So so clearly, there are fine-scale patterns of activity in ventral temporal cortex that distinguish these types of high-level visual stimuli. But we have a hard time modeling what are those patterns. So how can we model topography? And so I want to back up and talk first about how most people think about cortical topography, and that is they think about topography as a patchwork of functional cortical areas. And this goes back all the way to Corbini and Brodmann, who in 1909, so 109 years ago, published this map of cortex with 52 areas, and people still refer to Brodmann areas when they talk about human cortical and monkey cortical topography. Now, more modern versions use fMRI data and other sources, and there are now schemes with over 400 areas that people say are reasonably reliable. Now, the problem with this is that a cortical area, like let's look at this thing in the lateral occipital cortex right here, okay, is presented as just something that has an even color. In other words, there's no structure inside that cortical area. And in these kinds of schemes, that cortical area is characterized by what it does sort of globally. So when it's active, what is the brain doing? When it's not active, it's not doing that. But we know that these areas have finer grained topographies inside them. So let's start with V1. And is V1 here? Yeah. So V1's right down here. But we know that in V1, there are cortical topography topographies within that area. The first one is simply retinotopy. So from the calcarine sulcus, which has the representation of the horizontal meridian, to the border between V1 and V2, which is, has the representation of the vertical meridian, there is one cycle of change in the polar angle of the receptive field that's represented. Okay? But within that single cycle of retinotopic representation, we know that there are finer grained topographies. So this is work, also this is an fMRI generated map, or a map generated from fMRI data from the group at the University of Minnesota, and they mapped out where ocular dominance columns. So the columns that get input from the right eye are shown in red, the columns that get input from the left eye are shown in blue. And so you can see within this retinotopic V1, there is a map of ocular dominance. Now this scale of this is a 1 millimeter bar here. So these are about 1 millimeter across. And then there's a third topography superimposed or multiplexed with the retinotopy and ocular dominance, and that's orientation selectivity. So this shows orientation selectivity, again, measured with fMRI. Some people have doubts about this, but this is what, very much like what we see with using other methods. And you can see that this has an even finer spatial scale. So this is the key for orientation. And you can see within an ocular dominance column, so this red blob here is outlined with black here, and you can see all orientations are represented within that ocular dominance column. So we have a single cycle, spatial cycle of retinotopy, finer spatial frequencies of ocular dominance, and even finer spatial frequencies of orientation selectivity. And they're all superimposed or multiplexed with each other. And this isn't the end. There are other things that are represented in V1, a lot of other things that have their own topographies. So to think about V1 as just a functional area that's either turns on or turns off is a gross misrepresentation, and is really overlooking what is really interesting about V1. Now, when you go further in the visual pathway, let's go in the ventral visual pathway, people again talk about functional areas. So this is from Nancy Kanwisher's article in PNAS in 2010, showing the locations of the face areas in purple, the place areas in pink, and the body area in green, et cetera. And this is a more modern version of this, showing the extent in an individual subject of face areas in red and body areas in yellow and object areas in blue, et cetera. Now, again, these are functional areas, and they're characterized as simply areas with no attempt to look at what's inside these areas. But we know when we do multivariate pattern classification of patterns of activity that we measure with fMRI in an area such as the FFA, we can distinguish whether the person is looking at a human face or a dog face or a monkey face. We can distinguish if the person is looking at a human face in full profile, three-quarter profile, or full view. We can distinguish if it's this guy or this guy. Okay? We can distinguish, so this is all face information, but we can also distinguish, are they looking at a squirrel monkey or a ring-tailed lemur? And weirdly, we can even distinguish if they're looking at a shoe or a chair. So there is information inside the FFA encoded in fine-scale topographies that carries these kinds of distinctions. And we don't know what the basis functions for these are. We don't have things like orientation selectivity and retinotopy to analyze what is the basis of these topographies, but we know they're there. So can this information be modeled in a computational framework that's common across brains in the same way that we can say, okay, we have principles in V1, like retinotopy and orientation selectivity. Is there some way to find common bases across brains that carry information like, is it a ring-tailed lemur or a squirrel monkey? So to get at this, the first, we've been working at this for a long time. And what we finally came up with about 10 years ago, which really broke this thing wide open, is a new conceptual framework. So we model the information as a high-dimensional space. So instead of trying to line up the topographies, because it just doesn't work for those fine-scale topographies like orientation selectivity. There's no way you can warp one subject's orientation selectivity map to look like another person's orientation selectivity map and preserve the topology. You can do that with retinotopy. You can't do it with orientation selectivity. So instead, what we think about is if you have these patterns of cortical patterns here for different, these are T's for time points, you can vectorize these patterns, okay? And then you think about each one of these vectors, which is a cortical pattern, as a vector in a space. So this T1 vector is this location right here in a high-dimensional space. Each coordinate axis or dimension of this space is a single cortical location, which is usually a voxel or a vertex. So this is the critical step, is that instead of trying to find a common cortical space, we're going to try to find a common information space. So for stimuli, you can think of the pattern of response to the lemur monkey as being a vector with this location, or the pattern of response for a Luna moth as being a vector with this location. So these patterns, shown in the brain over here, all the information in the pattern is captured in the high-dimensional, in the vector in the high-dimensional space. It's the same thing. Now, I'm going to get into this later, but you can also use this framework to think about functional connectivity. So if you have a cortical area, like this lateral occipital area, it will have a pattern of connectivity with a target, a location elsewhere in the brain, like this area in right frontal cortex. And that pattern of connectivity with right frontal cortex is going to be different from the pattern of activity with central cortex or anterior temporal cortex. So each one of these patterns of connectivity in this cortical field is a vector and can be thought of as a location in high-dimensional connectivity space. So that's why I call this a high-dimensional information space, because it can accommodate response patterns. It can also accommodate connectivity patterns. And again, this is a radical departure. I don't know how many of you have worked with functional connectivity, but usually when people talk about functional connectivity, they talk about the connectivity between one big brain area and another big brain area. How much do they oscillate with each other? But we're talking really here about there's a fine-grained topography to that connectome. Okay, so this is how we're going to do it. So we start off with brains, and each brain has its own shape, size, and shape. And we convert a cortical space into an individual representational, individual information space, where each dimension is a voxel or a cortical vertex. Okay? Now these are idiosyncratic because voxel 2 in one subject's brain is not functionally aligned with voxel 2 in another subject's brain, even though they're anatomically aligned. Then what we want to do is we want to come up with some kind of transformation that will take these individual cortical information spaces and align them so that those vectors, those pattern response vectors or connectivity vectors, are in optimal alignment in a common information space. So let's start off by thinking about responses. We have people watch Indiana Jones, Raiders of the Lost Ark. And this is a scene early in the movie where Indiana Jones has just been bested by the evil French archaeologist, and he was stolen the golden idol that Indiana Jones had just stolen shortly before. So when people are looking at this, we assume because Spielberg is a gifted director, everyone's kind of in the same mental space, right? they're looking at the same things, they're having the same visual experience. And that in a visual area like lateral occipital cortex, the pattern of response is going to carry the same visual information. But because this 3-voxel lateral occipital area that's anatomically aligned is not functionally aligned, the vectors in their individual information spaces are in different locations. Now later on, Indiana Jones escapes on a seaplane, so people are having a very different experience, visual experience, but we assume that they're sharing their experience, and now we have different patterns of activity in the lateral occipital cortex, with vectors now shown in green in different locations. And you can see these vectors in subject 1 are in different locations from the vectors for the same time points in subject 3. So what we want to do is we want to somehow come up with transformations so that this green lines up with this green and this lines up with this green, and the reds line up in a common space over here. So there's the common space, and we want these to be clustered together. Now, if they're clustered together like this, then multivariate pattern classification based on a between subject classifier becomes much more efficient. So how do we do this? Well, as I said, we have people watch Indiana Jones and the Raiders of the Lost Ark. And so this is an early scene, and you can see that this is not like your standard controlled psychological experiment. Very cluttered, it's very dynamic. There's a storyline, that's Bullock, the evil archaeologist. And these are patterns of activity in 2 subjects' brains that are synchronized, okay, for the same time point. So whatever you see here is for the same time point as whatever you see here in the other subject, in ventral temporal cortex. And you can see the correspondence is not obvious. So this problem is not trivial, trying to line up these patterns with each other. So the idea is that as we see different patterns of activity, each pattern of activity is a vector in that subject's idiosyncratic cortical information space. Now this is just an illustration, but the red X's are the same time point, the green squares are the same time point, etc. Okay? So somehow we want to align this cloud of pattern response vectors with this cloud of pattern response vectors. So here they are, the pattern response vector for subject 1 and subject 2 for those 15 time points in a 3-dimensional representational space. Now keep in mind that with real movie data, we have 1,300 vectors for 1/2 of the movie, and we're dealing usually with a cortical field that has from 100 to 1,000 vertices, so that's 100 to 1,000 dimensions. So it's a much larger scale of what I'm showing here. So what we want to do is we want to come up with a transformation matrix, which is what's contained in these brackets, so we can multiply this data matrix by this transformation matrix and try to align this pattern with this pattern. And what we found is that if we derive a rotation, a rigid rotation, like that, using an algorithm called the Procrustes transformation, we get surprisingly good alignment of these vectors. Now, one of the things that's nice about Procrustes transformation, or the rigid rotation, is that it preserves the distances between all the pattern response vectors. So that representational geometry, which patterns are similar to each other, which patterns are different from each other, is preserved with this transformation. We think that's why it works so well. So we preserve representational geometry. So if we put these vectors now into the common space, you can see the time points are lined up with each other. Now, if we have a 3rd subject who has a completely idiosyncratic pattern of vectors, we need to derive a different transformation matrix for that 3rd subject. Okay? So the transformation matrices are specific to individuals. And when we calculate this using the Procrustes transformation and rotate the subject's vectors, now they're in beautiful alignment with the other 2 subjects. And now we have 3 examples of where the pattern vectors live in a high-dimensional common information space for each time point. And we can add subjects progressively like this until we get a large number of vectors for each time point, and we take the mean of that of those vectors as the location for that time point, or as the modal pattern response. So for those of you who like equations, it's very simple. This is it. We are deriving the common space, which is this matrix here, for and we're populating with the data for one subject, J. Is that what we're doing here? For one region, J, and that is the mean of the data for each individual subject in their native space times the transformation matrix R, okay? And we derive this transformation matrix R to minimize the difference between the individual's transformed pattern of vectors and the group mean using the Frobenius norm. Now, as I said, we use the Procrustes transformation in our work, but our friends at Princeton have derived other algorithms that can be used to calculate these transformation matrices, such as regularized canonical correlation analysis and probabilistic hyperalignment. And these also work well. So there's nothing about the Procrustes transformation that is dogmatic. And we continue to search for better ways of deriving these matrices. And then we have to put them into, we make a whole brain matrix by aggregating the transformation matrices for local hyperaligned models. I won't get into the details of that now. So does this work? So this is showing the math again in a different format. So this is the original brain data matrix. Voxels are columns, time points are rows. So each row is a pattern of activity. And we derive this transformation matrix, and it has voxels in the cortical area, and then the dimensions in the model, and the dimensions, the way we do it, is the same number as the voxels, but that's not necessary. And with that, we get this. We take this pattern of activity across voxels, and now we have the pattern of activity for the same time point across dimensions. We just move that vector to a location in a space with these model dimensions rather than voxels. So this is a pattern of response for time point 2. And this is the pattern of response for time point 2 in the model space. These patterns should be more classifiable than these patterns, as I showed you. Did it work? So it worked extraordinarily well. So this shows the between subject pattern classification of time points in the movie, or time segments in the movie. And after anatomical alignment, you can see we get classification rates above 5% only in early visual cortex. Now this is 1 out of 1,000, or actually 1 out of 1,300 classification. So chance is less than 1%, less than 1/10 of 1%. So this is still significant. But after hyperalignment, you can see the classification rates go up dramatically and now include lateral occipital cortex and lateral temporal cortex, parietal cortex, and prefrontal cortex. And to kind of get a sense of what these numbers are, rather than just colors on a brain map, we pulled out 20 functional loci. We have visual areas shown in red, auditory areas shown in blue, and cognitive areas shown in green, and looked at the classification rate in all these different functional areas. And you can see that the classification accuracy is much higher for the hyperline data, the blue bars, than the anatomically aligned data, the red bar, everywhere, including these areas in parietal cortex like the calculations area, and frontal cortex like expressive speech and working memory areas. Overall, it's about a 7-fold increase of classification accuracy. And this has general validity. So if we derive these transformation matrices based on the responses to the movies, okay, what I just showed you was properly cross-validated. So we derived the transformation matrices in half of the movie and then applied that to the other half of the movie to do the classification. Now with this, we took the transformation matrices derived from the movie viewing data and applied those to the data from this animal species category perception experiment. And you can see after anatomical alignment, classifications is pretty lousy, just in primary visual cortex and lateral occipital cortex. This is the within subject classification, so this is a map of what I showed you before. I just showed you a region of interest, ventral temporal. After hyperalignment, the between subject classification map looks essentially identical to the within subject classification map. So we solved the problem. We now get between subject classification that's equal to within subject classification. In fact, it's better. Now, how could that be? How can we derive a better model from other subjects than from that subject's own data? Well, that's because now we can take 12 subjects, or theoretically 100 subjects, to derive the model, okay? And so with an individual subject, we're limited by ethics, except in the Gallant lab, to how much data we can collect in an individual subject. And so the classification accuracy for within subject classification is constrained by the limited data set size. But between subjects, we don't have that limitation. We can just put one undergraduate after another into the scanner to get these data. Yeah? Yeah, so two things on that. First of all, we do some voxel selection because, well, it depends. Sometimes we do, but we do something to pick out the more informative voxels. Usually that's about 70% of the voxels. And it's important to note that we're not just aligning voxel. We're not saying voxel 1 is like voxel 3 in subject 2. So the data that goes into dimension 1 is a weighted sum of voxels in each subject's brain, okay? So it's not just matching voxels. Okay, now Luke asked me to make a few comments about what's special about naturalistic stimuli. So I dug these slides up from our first paper that documents this. So this again is this hyperalignment in this ventral temporal region of interest. And we can do hyperalignment based on the movie data. It's also possible to take the data from the category perception experiment, and derive transformation matrices from that. It's about the same amount of data. It's just that they're seeing still images of a small number of categories. So the question is that adequate to get these transformation matrices? And in fact, when I first started showing these results, I gave a talk at Harvard, and Patrick Kavanagh listened to the talk from Paris and said, why don't you just have people look at a few pictures, a few still images that are well controlled? And I said, because it doesn't work. And this shows why it doesn't work, okay? And so if you hyperalign based on the category experiment and use that to classify the data in that category experiment, it works really well. Those are the green bars here, okay? It works just as well as the transformation matrices from the movie. But if you take the category, the transformation matrices derived from the category experiment and apply them to the movie data, that classification of movie time segments crashes. It just doesn't have the general validity. It's very valid for the stimuli in that experiment, but doesn't have general validity across a wider, more representative range of visual stimuli. So that's one point. We get a much broader sampling of visual stimuli and brain states by using a naturalistic stimulus, like a movie. Yeah? The movie time segments, like I showed you in the last slide.
So yeah, because I kind of missed it in the last one. So what are you classifying with them? Because I guess all the segments are different.
So we're looking at, we can do this for any length of time segment. For that particular analysis, we looked at 15 seconds of movie data. We asked, can we tell exactly which 15 seconds of movie they're watching based on the patterns of activity that we measured during those 15 seconds, as compared to all other possible time segments in the movie. Now this is the one that's more interesting, and I hope this might inspire somebody for a hackathon project. So we said, okay, well, you know, what else is special about the movie? So what we did is we set up a classification where we picked out one time point in each run, a single time point for each of the 7 categories of stimuli in a face and object perception experiment. And then we took that same time point and looked at the response in the movie at that time point in the experiment. So we have 7-way classification from the object perception experiment, object category perception experiment, and 7-way classification from the movie data. Okay? So these are perfectly matched in terms of expected classification accuracy. And you might expect that these would be more classifiable because they're quite simple and and they're very distinctive from each other. But in fact, classification of the single TR of the movie, which is now 1 out of 7, is up around 75%, whereas for a single TR in the face and object is down around 37%. In fact, in other words, it's twice as strong, twice as accurate for the movie time points as it is for the face and objects. So, so that means that these movie stimuli are evoking patterns of response that are much more distinct from each other than those single still images. And why is that? Well, what I would like to argue is that there's something that is going on in a movie that controlled psychological experiments go to great pains to factor out, and that is prediction signal. So while you're watching a movie, you kind of know what's coming next. In fact, you really know what's coming next. So there's a very strong signal saying, okay, the next thing, you know, when I'm going to see, you know, in the next frame, there's still going to be Indiana Jones and Bullock, right? And, you know, in the predictive coding framework, that plays a very important role in making visual perception more efficient. So the idea is that there's a very strong neural signal predicting what's coming next for filtering, interpreting input, right? And with a movie, that prediction signal is very meaningful and shared across subjects. In the category perception experiment, there's almost no prediction. You know it's going to be 1 out of 7 things. You don't know what it's going to be before it comes up. So there isn't a meaningful shared prediction signal, even though that might be a large part of what the neural activity is in an experiment, is making predictions about what's coming next. So this huge increase in classifiability may be because we are now able to also exploit a meaningful shared prediction signal across subjects. Yeah?
Sense for how much variability there is associated with individual versus TR. So like this result could theoretically be due to either like the subjects could still look more different from one another during the movie as long as the movie time points are also really, really dissimilar from one another in this space, versus like the category stimuli could be like there could be less individual variability to the category stimuli, but the stimuli themselves are also closer together in the space.
Yeah, I mean, if there's less individual variability for the category experiment, there should be higher between subject classification.
Right.
Not lower.
If, like, the stimuli themselves are still separable. So, like, I guess what I'm getting at is, like, the ratio of category or TR variability to subject level variability. Like
Yeah, I mean, we're showing that the between subject variability for the TRs is actually quite low, as shown by this kind of high classification rate. And we have to explain that. Now, it could be prediction signal. It also could be because we actually, the richness of the stimulus, the dynamic motion, everything, is playing a role in making it a distinctive time point. So this isn't definitively evidence for the predictive coding playing a large role in the responses to a movie. But my prediction is, okay, that when the prediction is weak, classification accuracy should go down. So when is the prediction signal weak in a movie? Well, it's when there's a change of scenery. So when Indiana Jones is dealing with Balak and the seaplane escape at the beginning, then I think right after that it changes to a classroom at University of Chicago or Princeton University or something like that. So it's completely unexpected, disorienting change of scenery. At that point, those first couple scenes of those academic buildings and the classrooms should be less classifiable because there's no meaningful prediction signal. So this is a hypothesis that could be tested with the existing data. So just throwing out there in case you're interested. Pardon me? Yeah. Yeah, and so there's work by Chris Honey that you'll be hearing about later on, where they scramble the movies up to make it less predictable from one, so to break up that kind of structure that affords good prediction. So that's another way of looking at it. Yeah. Okay. Now I'm going to get into another way of looking at these data, which is a way of looking at, rather than time point by time point, it's looking at the whole representational structure for the movie. And the way you do that is using something called representational geometry, or representational similarity analysis that Nico Kriegeskorta is really responsible. It was played a key role in introducing for fMRI data analysis. And so we're really looking at the whole data set, either before or after transformation. And what we're looking at is how similar is the pattern of response to time point 1 to the pattern of response at time point 2, time point 3, time point 4, et cetera. Okay? So you can imagine that two time points that both have Harrison Ford in them might be quite similar in visual areas, and a time point where there's Harrison Ford in one segment and a mountain scene in Nepal in another might be very different from each other. Or in an auditory area, you might have two time points where the same theme music is playing, and they're going to be very similar, whereas a time point with theme music versus Marion talking would be very different. So you have this kind of very complex representational geometry for the movie content. And so we do this for all time points. So for half of the movie, that's a matrix of pairwise similarities that's 1,300 by 1,300, or about 800 pairwise similarities. And we ask, how similar is that representational geometry across subjects? And we can show that inter-subject similarity of representational geometry is greatly enhanced by hyperalignment. Okay? So hyperalignment is somehow getting the information in a searchlight to be more consistent across subjects by resampling information from the neighborhood into that searchlight, or sampling information out of that searchlight that is not shared across subject for that area. So this is a big effect. But we went ahead and then we did something different. And that is we looked at what we call second order representational similarity analysis. So that is we look at the representational geometry for one brain area, like a visual area, the FFA, on the left, and say how similar is that representational geometry to the representational geometry in other visual areas, or in the auditory areas or the cognitive areas. So this is a matrix of all pairwise similarities of representational geometry in different brain areas. And you can see there's some nice structure to it. First of all, V1 is different from everything, but the visual areas are most like visual areas. Auditory areas are most like auditory areas. and visual and auditory areas are very different from each other. And then there's this interesting structure down here. Now, it's very hard to digest a 20 by 20 similarity matrix like this. So one way to try to get at this is to put it into a multidimensional scaling plot. So make it a lower dimensional plot. And that's what we have here. So you can see that in this three-dimensional, multidimensional scaling plot, dimension 1 pulls visual areas apart from auditory areas with the cognitive areas green in between. And then dimension 2 pulls V1 apart from everything else. And then dimension 3, over here on the right panel, distinguishes the cognitive areas from the sensory perceptual areas. So we see sort of the overall structure, functional structure of the brain for these functionally defined loci from the second-order representational geometry. Now, this can be used in other experiments. So Matteo Visconti di Allegro Costello and Ida Gobini did an analysis on their data, and then they reanalyzed the movie data using this, looking at functional loci in the face pathway. And so you can see there are early visual areas shown in blue. There's the posterior core system shown in red, the anterior core system shown in orange, and the theory of mind and precuneus here. And you get this beautiful structure showing the flow of information through the face system from the movie data based on representational geometry. Okay. So, I'm running out of time, so this is just an analysis of what is the spatial granularity of this common model? You know, is it true that we're getting down to fine-grained patterns, or is there something else that's really driving it? So we did an analysis, we did a couple of analyses, and one of these is this point spread function, where we look at how similar is a given cortical vertex with neighboring cortical vertices, with itself and with neighboring cortical vertices in other subjects. And we get a much sharper focusing of the functional signature across cortical distances in the hyperaligned data than in the anatomically aligned data, showing that we really are picking up differences in the functional signature for neighboring vertices. So we're down to about a 2-3 millimeter granularity. Okay, so this is when we apply this to connectivity. So now we use the same math, but we apply it to these connectivity data matrices. And we're looking at, in this one example I'm going to show you, we look at the connectivity profile for a voxel, and we compare that to the connectivity profile for a dimension. And we look at the between-subject correlation. So how similar is the connectivity profile for voxels in the anatomically aligned data, and how similar are the connectivity profiles for voxels or cortical vertices in the hyperaligned data using connectivity hyperalignment. And you can see that now we get a really dramatic increase in the similarity of the connectome across subjects with a very sharp point spread function, showing that the connectivity profile for a given cortical vertex is significantly different from the connectivity profile for neighboring cortical vertices. And the advantage of this is now we don't need to have people watch the same movie. Not everybody likes Indiana Jones. We might want to compare adults to children. And there are some movies that children shouldn't be watching, right? So, but now we can look at connectivity rather than synchronized responses to a stimulus. And that means we can even look at resting state data. So in this paper, we took data from the Human Connectome Project, resting state fMRI data. We just took a small sample of subjects, 20 subjects, and showed that the between-subject correlation of connectivity profiles is dramatically improved after we do connectivity hyperalignment. Okay, and now I want to get into Fei Long stuff on individual differences. There's Fei Long. There's Fei Long. And what Fei Long did is he developed an index of how similar two subjects are to each other in terms of their cortical fMRI data. And to do that, he would look at the pattern of response in a searchlight, okay? And he would vectorize it. So we have one long vector for all time points, all cortical vertices for subject 1, and then we have another vector for with the same time points and the same cortical vertices in another subject. And you correlate these two, and that's a measure of how similar is subject 1 to subject 2. And so you can make now a matrix of differences, but now this is individual differences in their cortical responses or cortical indices. And after anatomical alignment, you can see that subjects are fairly different from each other, okay? But after hyperalignment, they're more similar to each other. That's what we'd expect. That was the whole point of hyperalignment, is to make subjects more similar to each other. And so one might expect that the individual differences after you hyperline the data will be kind of noisy because you factored out everything that's similar. You factored out what's similar across subjects. But by contrast, what we find, so first of all, he looks at the reliability. So how much is this matrix derived from one data set similar to the individual differences matrix derived from independent data, anatomically aligned data, and for hyperaligned data. And we get quite high reliabilities. So this is a map of reliabilities of these individual difference matrices in anatomically aligned data. You can see that these individual difference matrices are very stable in these auditory and visual areas, parietal cortex, and this prefrontal cortex. Not so much in anterior prefrontal or in medial prefrontal, and not in sensory motor cortex, which you'd kind of expect because they're lying still. After hyperalignment, so we might expect these reliabilities to go down, but in fact they go up, dramatically up. And these reliabilities are now highly significant in interesting places like medial prefrontal cortex and anterior prefrontal cortex. So we're getting reliable individual difference measures across the cortex that are much better after hyperalignment. So this is just a scatter plot. Yark's not here. Yark loves these scatter plots. So these are the reliabilities after anatomical alignment, and these are the reliabilities after hyperalignment for searchlight. So each dot is a different searchlight, and you can see that everything, almost everything, is on this side of the identity line. So then what Fei Long did, which really solved this mystery of why are these individual differences more reliable after hyperalignment, is he divided the data into data that is related to coarse scale information and data related to fine scale. So for each searchlight, he got the average time series or the average connectivity profile, okay? But in this, it's the average time series, and looked at the similarity between subjects in terms of that single time series. So that's the core structure for the whole searchlight. And then he subtracted that average time series out from all the individual vertex time series. So now each vertex has a different distinctive time series. That's the residual. And that's the fine scale information that varies voxel to voxel within a searchlight. So what you can see here is that the reliability for the core scale information is about the same for anatomically aligned and hyperaligned data. There's really no advantage. And this is the scatter plot showing that result. The fine scale information, though, is much more reliable after hyperalignment than after anatomical alignment. And this is the scatter plot. So what this is doing is it's capturing individual differences in the information encoded in fine scale topographies, okay? And finding reliable measures of those individual differences. So the next question is, do the individual differences mean anything? Or is this just a nice statistical demonstration? So he went back, and these are from movie data, but Fei Long went and analyzed data from the Human Connectome project and tried to predict performance on a psychometric test, picture vocabulary, which is a test of verbal intelligence, based on patterns of connectivity, on this individual difference matrix derived from patterns of connectivity in different searchlights. And he got a nice, significant result with anatomically aligned data. After hyperlining, these predictions went way up. So what that suggests is that the individual differences in cognitive ability that are reflected in psychometric tests like picture vocabulary are based on individual differences in how information is encoded in fine-scale topographies, much more so than in coarse-scale topographies. And if you follow the literature, individual differences in fMRI, it's all on coarse-scale indices of between area connectivities, or the strength of response in an area, or the size of a cortical area. But this suggests that really, people are looking where the light is, but the keys aren't there under the light. The keys are someplace else, and they're in these fine-scale topographies, and the information encoded in fine-scale topographies. So I'll do a quick summary here. So the basic components of the common model are, there's this high-dimensional space that you can have both information about responses, information about connectivity in this space, and transformation matrices that are specific to each individual that project individual cortical spaces into the high-dimensional space. And I haven't shown it, but you can also project data from the high-dimensional space back into individual topographies. And the algorithm that accomplishes all this is called hyperalignment. The dimensions are very complicated things. So they carry a lot of information. They have response tuning basis functions that are common across brains. Okay? They also have functional connectivity vectors that are common across brains. And I didn't show this, but they also are associated with topographic basis functions. So topographic patterns that are idiosyncratic to each individual, with which we can model individual topographies with very high fidelity. So the fine-scale structure is really what this is all about. It's a big factor, 7 times higher between subject multivariate pattern classification, greatly increased inter-subject correlations of response tuning profiles, local representational geometries, connectivity profiles. So we call this the dark matter of cortical functional architecture, something that people weren't able really to look at before with previous analytic methods, because we couldn't get a common model of what this fine-scale structure was. I think I want to go to, and then individual differences. This is what I'm really excited about these days. Hyperalignment increases the reliability of these individual differences by capturing variation in the deep structure of information that's not accessible in anatomically aligned data. And these individual differences better predict cognitive differences. So the granularity of cortical architecture that underlies cognitive differences is matched better by the scale of fine-scale topographies than by the coarse scale of systems of functional areas. That's the take-home message here. And the second take-home message is show these guys some respect. So this is Jarek, 9 years ago. There's Baker Tower behind him. And Michael Hanke, who was here for a couple of years. He's now a professor at the University of Magdeburg in Germany. And this wonderful suite of software that they've been developing, Pine VPA, NeuroDebian, DataLat, he talked about this, and his Center for Open Neuroscience. And then these are just some references for some of the more recent papers. So there's this paper by Swarup on whole brain hyperalignment, another paper by Swarup on Human Connectome, on connectivity hyperalignment, and then a paper by Fei Long that is now on bioRxiv. We hope it'll be published soon on the individual differences. Thank you for your attention. Andy.
That's a great talk. So I still don't understand how to calculate fine scale thing. You
Oh, in Fei Long's analysis?
Yeah.
Okay, so for all the vertices in the searchlight, there's an average time series. Okay, so that's one time series for the whole area. That's the coarse information of that area. And then you take that time series and you subtract it from the time series for each individual vertex. So each individual vertex has a different time series. And so the residual varies, the residual time series varies vertex by vertex. So you have a fine scale pattern of variation in the time, in the time series response after you factor that out. So these are independent. So you have the, you divided the variance into the shared variance across all vertices and the idiosyncratic variance for each vertex. Yeah.
The difference between the notation method you used for hyperalignment and simply PCA?
Well, when we started this, we didn't use PCA because there's a lot of variance in fMRI data sets that is not interesting. That has to do with people heartbeats or when they move their head, okay? And so if you do a PCA on each individual subject, the top components are uninteresting things like that. So that doesn't work. Now there's another way of doing PCA, which is called joint PCA or joint SVD. So each subject's data is a table, and you do PCA across that table. And we've tried that too, and the results just aren't as good as this. And we're not exactly sure why, but it just doesn't work as well. Yeah. Yeah, so you can see that, first of all, the classification rates and alignment of functional data is much better in prefrontal cortex after hyperalignment. And the functional connectivity is dramatically better. So we're doing a much better job aligning functional connectivity in prefrontal and medial prefrontal cortex. But, and I think that you should try to relate this to what Chris Honey's going to talk about later in terms of time scales, that the responses to the stimuli are quite similar across subjects, whereas how people think about that starts to vary. And so, so higher level representation of what is the meaning of the stimulus, is more idiosyncratic, and especially in default areas like medial prefrontal cortex and anterior lateral temporal cortex, we expect that to be much more idiosyncratic, and that's actually one of our primary motivations for developing the connectivity hyperalignment algorithm, because we thought that the kind of information they're representing in these higher order areas is idiosyncratic. But the way these higher order areas are communicating with the rest of the brain may be more similar across brains, and that's kind of what we're finding. Yeah?
Overlaid maps of spatial and temporal SNR over your group hyperalignment maps. How much variance would you think that some of that sort of like uninteresting, maybe scanner-driven variance is driving accuracy?
Yeah, that's an interesting question. So, I mean, we know that we have a lot of trouble in areas that have dropout because of artifacts like susceptibility artifact. And if the data are worse, the results are worse, right? But the surprising thing is that these maps show pretty strong results in the areas where we have good signal. It's pretty consistent everywhere, except in areas like sensory motor cortex, around the central sulcus, because subjects are lying still. Yeah. Carolyn. Yeah.
One question, and I think that makes sense based on the point that you brought up of the static stimuli being devoid of context
Okay, well, so we've done that in a way that people don't find completely convincing, because we derive it on one half of the movie, and then we apply it to the other half of the movie. All of our cross-validation is doing that. So that's a different naturalistic stimulus. There's different things happening in the second half of Indiana Jones than the first half. We've also done this with a nature documentary, and there are different animals and different animal behaviors in the second half of the documentary than the first half. And we actually have enough data, and we haven't tried it yet, to go between movies. So if we derive the matrices on a Hollywood movie and apply it to a David Attenborough nature documentary, we haven't seen how much loss of commonality we have there. But I think we have enough data, we have to actually do that kind of thing. There's another hackathon project. Bo.
I just briefly want to speak to that. It works pretty well.
Okay. So Bo's going to have his defense on August 31st, and so you can we'll see it then. Yeah.
Somewhat related to that, have you think about how much time you have to actually collect?
Yeah, we've done that. I have a slide somewhere in here that so we've derived the model based on 10 minutes of data, 20 minutes of data, half an hour of data, and when you get up to 20 minutes of data, it's already pretty good. The more data, the better, but the incremental improvement from 20 to 30 to 50 minutes is smallish. So you can probably get away with about 20 minutes and get decent results. My policy always is collect as much data as we possibly can, and then we'll try to make things efficient later. That's how we do it with computational algorithms too. I say, just do everything in the computational algorithm, and then we'll see if we can, make it efficient later. But first, we want to see if it works. Pardon me? With 8 hours?
No, is it like would you need like less data for getting like the same good results, like or?
For the connectivity alignment.
Hmm?
Do you need as much data for the connectivity alignment?
That's another thing that we haven't really analyzed carefully, because it looks like the connectivity may be more efficient in terms of because each vector in the connectivity matrix is really deriving data from the whole brain, because it's that voxel and all the other voxels in the brain. So we think that it's a much less noisy data set in that sense. But we haven't done that analysis, but it might be more efficient. It's not perfect, though. Connectivity hyperalignment does a really good job aligning connectivity and does a pretty good job aligning responses, but we do a better job aligning responses with response hyperalignment. So if we do connectivity hyperalignment, if we look at connectivity in the response hyperaligned data, it's pretty well aligned, but it's better aligned if we use connectivity hyperalignment. So yeah, there's room for improvement that way. Alex.
So I really like the point that you made early on about the difference between like visual areas versus the like sub-visual areas,
Right.
ocular dominance patches or orientation patches. It really seems like one of those is kind of genetically encoded, like everybody has a V1, everybody has a V2, et cetera. And the other one is maybe learned, or there's some complex like developmental process that gives us orientation. Like there's no reason to suspect that everybody is going to have the same orientation, the same set of orientation columns, and there's a one-to-one mapping between them, right? And I feel like that's a nice sort of division between the things that might be large-scale structure, the things that might be fine-scale structures, the things that are like genetically, whatever, like coded in some.
I'd be very cautious there,
sure,
but.
Yeah. But things that have some sort of that are encoded in some way that's not specific to a person versus things that are not. But how do you square that idea with the fact that like the hyperalignment seems to work differently in different parts of the brain, right? So as someone pointed out earlier, like in sort of early sensory areas, the anatomical alignment works decently well, and hyperalignment adds a lot on top of that. But then in like prefrontal cortex, anatomical alignment doesn't do anything,
Right.
essentially. But hyperalignment like brings out these things that are shared. Do you think that there's just less of these sort of large-scale common patterns, or what's happening?
Yeah, I think, well, in prefrontal cortex, the large-scale patterns are really a mystery right now, right? Yeah, and these suggest that anatomical alignment is not really aligning anything meaningful in prefrontal cortex, even though we're showing that there's a lot of meaningful information that can be drawn out if you think about it in terms of an information space rather than a cortical topography. I don't have anything more to say on that. I'm sorry, I can't be more intelligent.
Last question.
Quick comment on that. I think, at least not so much with hyperalignment, but with other resting state data that I've kind of played with it, looking at reliability of individual differences is going to play quite a role in that. So if you're looking at more kind of, visual, somatomotor regions like that, those regions are pretty similar across people, but once you start looking at like frontal or things like that, then you end up getting larger, reliable individual differences. So that, if you're doing a rotation, might not work very well pulling, if you're trying to rotate something that's actually a real individual difference versus something that's just a different area.
All right, really.
Rob.
Question, comment. So isn't a lot of this dependent on the type of stimulus you align to? So instead of Indiana Jones, you presented people with something with a lot of emotional content. Would you expect emotion-related brain regions
Yeah.
to be?
Yeah, we haven't done that systematically. We have done, a Hollywood movie with human actors, and we've done a David Attenborough movie with only animals in it, because we do a lot of work on representation of semantic knowledge about animals. And we want to compare whether or not the, nature documentary does a better job aligning information about semantic representation of animals than Indiana Jones, which has almost no animals in it except for a poor monkey that was poisoned. But yeah, so I think that if you're interested in something like emotion or making inferences about someone's mental states, there are almost no inferences to make about mental states in Indiana Jones, right? Because they're bad guys and good guys, and it's pretty simple. And so you might be able to do a much better job if you had a movie that really made people think about what people are thinking about. And Uri Hassan has done that, to a certain extent with his movie Bang You're Dead, that Hitchcock episode from the 50s, where everyone's thinking, is this kid, nobody knows this kid's carrying a real pistol with live bullets in it. And so everyone's thinking about what people are thinking. And he gets really good synchronization of activity in like medial prefrontal cortex with Bang You're Dead, whereas you don't get it with other kinds of movies.
All right, great. So let's take a 15-minute break and thank Jim one more time.